without further ado, then I think we should dive in to the content of this very exciting webinar. I'm extremely looking forward to seeing what Frederik has to say. So I'd like to hand over the floor to Dr. Frederik Zitzmann, who did his PhD in green chemistry from York and Algaecytes. He's now a research coordinator at Co2Co. So I think we're going to see throughout this presentation, you know, how really academic findings and research is really translated to industry. And now he's heading very, very soon over to Dorowood to continue his work there. So without further ado, welcome Frederik to the stage. The floor is yours.
Thanks very much. Hi, I'm Frederik. Like I've been introduced just now, I have done my PhD at the University of York Green Chemistry Centre of Excellence, and I've worked together with Ebba Biotech on multiple occasions during my journey there, during my PhD, and it's helped me a lot. And that's why we're all here today to talk a little bit about how Ebba Biotech and Carbotrace in particular have helped me during my research and really helped me make big steps towards reaching my goals during my PhD. So here we go. So today's all going to be about incorporating Carbotrace as a problem solver in a microalgal biorefinery design process. So here's a little bit about me. So between 2019 and 2023, I did my PhD at the Green Chemistry Centre of Excellence at the University of York in the UK under Professor Aftar Matharu in collaboration with Algaecytes. I'll talk a little bit more about Algaecytes a little bit later. Basically, I was doing a semi-industrial, semi-academic PhD with industrial collaboration. I already did my master's with them prior to the PhD, and it's been sort of like a 5-year continuous project, which has been very exciting. After that, I've been working for Co2Co in London for a few months as research coordinator, also working in the microalgal field, basically looking at microalgal projects, trying to, you know, diversify the product range and the application range for microalgae in industry and being at the forefront of innovation there. And then very excitingly, from August onwards, I'll be working in Austria for Dorowood, bit of a change in scenery. So I'm going to be working in the wood additive and resin industry. So I'm going to be transitioning a little bit. Just a short introduction on microalgae for people who might not have been involved with them before, or who might not have heard much about them. Basically, they're very interesting microorganisms. They're unicellular, photosynthetic. They exist all across the globe, pretty much everywhere. It might be in oceans, in rivers, in lakes, in hot water pipes, in pots outside in your garden if you leave it out for too long, basically anywhere. They're very versatile. There are more than hundreds of thousands of species out there. They're very diverse. Not just in terms of their composition, but also in terms of just their physical appearance. As you can see down there, there's a few examples of what the cells look like. They really exhibit such a diverse profile, and lots of them are really not very intensely researched yet. So there's a few products and few species that you might have heard of before if you've been walking through the supermarket aisles. For example, chlorella and spirulina and nanochloropsis, they're kind of the very popular ones out there that you can see in the supermarkets nowadays as powder additives, superfoods, whatever you want to call them. But basically, they're much more than that, and they have so much potential in terms of biomass sources and product sources. It's quite astonishing, actually. So basically, what you can do, you can grow the algae. They grow via photosynthesis. You cultivate them. You don't really need to add much. Basically, they just need sunlight or LED sunlight. They need some nutrients like phosphates, nitrates to grow, and they need carbon dioxide for their photosynthesis. And then they can grow very rapidly in highly efficient growth cycles. Then you can harvest them, you can dry them, or you can use them wet, however you want. You can use them in a microalgal biorefinery to get all your different biorefinery products. And then from the released CO2 of all those processes, say through biofuels, etc., you can then feed that carbon dioxide back into the microalgal cultivation. So you've got a great sustainable circle. And it's one of the greatest photosynthetic organisms out there. They're really versatile. They produce around 50% of global oxygen. So they are kind of the unsung heroes of the plant world. Everybody knows about trees and plants. Not that many people know about microalgae. but they really are valuable. They've got lipids, proteins, antioxidants, pigments, carbohydrates, and many more. So they're very versatile species. Basically, what you can do in terms of proper product applications later on, and up here you can see some images of my microalgae that I've used. So they basically look like matcha tea powder, if you know the Japanese teas. They almost smell the same, a little bit more fishy, but essentially they look the same when you dry them. You can really take the composition profile. In this case, this is the profile of the algae that I've used myself. So you've got pretty much half, like 41% proteins. So it's very rich in protein, especially this strain I used. It's got lots of lipids as well, and carbohydrates, and lots of antioxidants. So you can really gear and tune them towards anything that you're trying to make, ranging from pigments, peptides, polyunsaturated fatty acids, omega-3s, food supplements, food additives. You can use it for the cosmetics industry, nutritional industry, vegan proteins. You can make biofuels out of it. You can make biochars for chemical absorbance, for wastewater treatment, and all these kind of things. So basically, the world is your oyster. You can do anything you can think of. You can tailor them. So they are a really great source of biomass because they're easily tailored toward any kind of application. And they're very exciting to work with. So I've always had fun over the last 4 years working with them. A little bit about Algaecytes itself, the company that I've been collaborating with. So Algaecytes are located in Kent in Southeast England. And they're also, there's the pilot plant in Germany, which is currently under planning and construction phase. So Algaecytes basically cultivates and harvests their own proprietary microalgae. So down there you can see some of their laboratories where they have the tubular reactors where they basically grow their own microalgae. So you can see the pink LED light, which is being used to optimally grow them. And then in the left, you can see the harvesting tanks where the algae are being collected before they're being centrifuged, dried, and then used for extraction. And one of the main products of Algaecytes is the omega-3 powder. So the algae are very high in EPA. So basically, it's a vegan EPA omega-3 acid, which usually is being sourced from fish or from salmon. So to cut out basically the salmon farming and avoid the unsustainable fishing, you can use microalgae to produce your omega-3s. And you can basically cut out the middleman and start producing omega-3s directly from the source, also from the marine source where the fish would get them from. So basically, Algaecytes are cultivating their algae, they're extracting their EPA, their omega-3s, they're selling the oil, and they're left with lots of biomass waste. And especially with the proposed biorefinery plant in Germany, which would upgrade the current 16,000 litre facility they have in Kent up to 2 million litres. So with that increase in capacity, there's obviously an increased biomass waste associated with it as well. So that's where I came in 5 years ago when I started my master's. So basically look at this biomass waste and set up and create a biorefinery process to valorize this biomass waste, I'll call it, and generate products from it. So that's been my journey over the last 5 years, which extended then into the PhD project. Has been very exciting. And we've reached a lot of goals together and managed to really create a biorefinery circle with lots of different cool products. So this is kind of like a brief overview of the biorefinery cycle that we designed in general. So we're basically starting from the spray-dried algae. Spray drying is just basically you take the algae out, they're being centrifuged, and then they're being spray dried. So they're basically just being pumped through a nozzle, which is heated, and you can imagine it like a can of deodorant, something like that. And then you spray the water off, and your dried powder falls to the floor and is being collected. So after the algae have been dried, they are being used for the industrial EPA extraction, and this EPA omega-3 acid is then sold to industry. The spent waste biomass has then been shipped to my laboratory at the University of York, where I did lots of different things on them in the laboratory. One of them has been the extraction of vegan peptides using microwaves and membrane filtration. So I've always been trying to avoid any sort of like chemicals and and also organic solvents to keep it as green and circular as possible. So we use just water-based membrane filtration, which has been working very well to extract and purify peptides, which we've then formulated for application in things like cosmetics, sport drinks, or nutritional supplements. With the residual waste biomass from that protein extraction process, which is getting less and less each time, we've then done a couple of different things. So one of them was the microwave pyrolysis to generate biochar and bio oils. Those can be used as catalysts for chemical reactions. So you can imagine the biochar as a heterogeneous catalyst which catalyzes different chemical reactions. The char also has lots of pores on its surface, which can be used for adsorption of different molecules, which can be used in the wastewater treatment industry, for example. And your bio oil serves as a biofuel or as platform molecules for further synthesis. The interesting part for this seminar, and also for Ebba Biotech, is the second thing that we did using solvent washes to afford defibrillated cellulose fibers. These defibrillated cellulose fibers can then be used for hydrogel formation. So you can imagine you're in a medicinal setting and you use a hydrogel, which can be used for wound healing, for example, applying to the body. And you can also use those fibrillated cellulose for bioengineering. You can make fibers from it, you can make textiles, you can make different microengineered products. And that's really the point where the Carbotrace came in and really kind of saved the day, saved the research in that sense. So I'll be talking about that part of my research in depth later on. Just a quick overview about the other kind of side streams that I did from the microalgae. The big part of my research has been the proteins, especially the vegan proteins. Vegan protein market is growing rapidly, and lots of people are searching for alternatives for proteins from, turkey, beef, egg, milk, these kind of plants, kind of animal sources, and also from plant sources like soy and chickpea, because you've got this food versus usually it's called food versus fuel debate. In our case, it would be a slightly different kind of thing. But, if you're looking at products in a biorefinery sense, you're trying to avoid as many competing food sources as possible. And as the proteins are kind of like a byproduct, we're trying to, go as vegan as we can. So the whole process in my case was using the microalgal biomass and then either pre-treating it or not. The pre-treatment comprises methods such as ultrasonication, just manual grinding, enzymatic treatment, but also microwaves. And that's just a very efficient way to break up the cell walls and facilitate the extraction and purification of proteins. This step is then followed by centrifugation, membrane filtration to separate your proteins from all the carbohydrates, cell debris, and everything else that might be floating around, and then to really purify your proteins. And down there, you can see the protein content of several microalgal species, and you can see that a lot of them are quite high and sometimes even higher than conventional sources like turkey, beef, or vegetarian sources like soy. And on the left side here, you can see the amino acid split of ALG01, which is my microalgal species that I used, compared with the amino acid composition of the soy. And these are the essential amino acids. And you can see that pretty much every amino acid, apart from cysteine and histidine, are present in a higher quantity in the algae compared to soy, which is also pretty good. And we did manage to get really highly purified and bioactive anti-inflammatory proteins out, which we then proposed to be used for cosmetics and nutrition or medicine even. Using microwave technology, I also used pyrolysis to afford biochars and bio oils. In terms of the biochar, like I mentioned earlier, you can use them as biofuels, for example. You can see an increase in the higher heating value of the biochars compared to the initial biomass before the pyrolysis. So you can get an energy densification there. You also in the bio oil get lots of different organic acids like myristic acid, palmitic acid, stearic acid, etc. Even vitamin E in the bio oil, which you can then use as products or as platform molecules, or use them in the chemical synthesis, whatever you're desiring to do. So you really have lots of choices with your different products from the algae, what you want to be doing. The interesting part now for us and Ebba Biotech was the defibrillated cellulose. And that's also been the part of the research which has kind of led me most into uncharted territories, where I came out at a different stage where I was initially proceeding towards, and which I expected, which was very interesting, because research often thrives from it leading you into new directions and discovering new areas which you wouldn't have guessed you would explore before. So just a general overview of what I've been doing. I've been, again, using my algal waste from the industry, and I've been treating it in this beautiful machine over here, which is a synthwave microwave, which looks very scary, which basically just kind of like the normal household microwave, refined a little bit towards academic and industrial application. Just basically heats your biomass under pressure in water, so it almost reaches subcritical conditions, and it basically heats up your biomass water mixture at temperatures between 160 and 220 degrees. Those were the temperatures that I used. After treating the biomass at these temperatures for 15 minutes, I centrifuged it, and I generated two fractions, the aqueous supernatant and the solid pellet. The aqueous supernatant was then used with membrane filtration to generate the proteins and peptides that I've been talking about before, basically giving us the precursor for formulation in the cosmetics industry or the food industry. And the pellet was then washed with a few solvents like water, ethanol, and acetone to generate defibrillated cellulose fibers. Basically, what you're doing is, through the heat treatment and the solvent washes, you're washing out all the other components like hemicellulose, xylan, lignin, whatever might be still in there, and you should be left with pretty pure cellulose. At least that's what we've seen from previous PhD students in my research group. We've had a big focus on these kinds of defibrillated cellulose fibers from plant material, so orange waste, ginger peels, black currants, these kind of things. And that was the first time for us that we've been using marine species and microalgae. So we were going in with high hopes, because things have been working very well previously. And we were basically planning to see defibrillated cellulose fibers. So you can see here at the 180 degrees, it's like a multi-lane highway where you've got lots of long parallel running cellulose strands. And as you increase your temperature to 220 degrees, you're starting to kind of break apart the fibers a little bit more. You're kind of starting to cut them off, and they're getting a bit thinner. And then finally at 220 degrees, you're really defibrillating the cellulose fibers. They're getting much thinner, and they're getting cut up, and you can even see some onsets of nanocrystals. So that's what we were hoping to see. But when we were analyzing these cellulose fibers further, and also when we were basically trying to form hydrogels, we were running into a few problems. So one of the first problem was, when comparing to cellulose hydrogels of previous PhD students and colleagues, they usually form beautiful gels. You homogenize them, basically mix them with water, you homogenize them, and you're left with a really nice thick gel, which upon inversion of a sample vial would stay at the top of the vial and be really thick and flow down. So it would be a brilliant gel, and you can use it for different applications, modify it and everything. But when I did it, I found that it was very, very tough to form any gels. They were very short-lived when I managed to generate them. And most of the time when I generated them, they just, it was just the 220-degree sample. So the sample which has been treated at the high temperature for a long time, which managed to get a little bit of a hydrogel. So we were thinking, why is that? Do we see impure cellulose? Like, what's the reason for it, when previously with other biomass it's been working so well? And we've been given basically contradicting evidence from the different analytical methods that we shot at the samples. So we used, for example, the TEMs like I showed you earlier, and also infrared IR spectroscopy and solid state NMR, which is the spectra you can see here, which are usually very conclusive when you're looking at cellulose and these kinds of structures. So ideally, up here, you can see the pure spectrum for cellulose. That's basically what the 60 to 110 ppm region should look like if you have pure cellulose. Down here, you can see my samples. On the left side, you've got spray-dried algae. On the right side, you've got spent algae. Don't need to worry about the terminology too much. It's just two different types of microalgae from two different stages in the process. And I've been constantly comparing the two with each other. So this pure cellulose region here would correspond to the region down here, which I've labeled according to the number of carbons in the glucose rings of the cellulose. And if you compare them, you can pretty much see straight away that we are really not replicating the pure cellulose spectrum in any way. You do get some overlaps in terms of the peaks. So these broad peaks here and that peak around like 107 ppm over there, which you can kind of assign to the spectrum, but you're very broad and the peaks are not very clearly able to be assigned. We do have, with increasing temperature, like the two arrows here indicate, you do see some sort of increase in the crystalline cellulose, but again, it's not very conclusive. We've got lots of other peaks in here. I've labeled them algaenan, which I'll explain later, which we weren't really sure what they were before. So basically, the question we were asking ourselves, there should be cellulose in there. We can see it from the TEMs, but in the spectra and in the hydrogels, we're not really seeing conclusive evidence that there is cellulose. And if there is cellulose, there has to be something else in there. And how pure are we? Do we really have pure cellulose? How does the cellulose spatial distribution in our samples look like? We were basically clueless. And we've been trying so many different analytical methods that we were really at a loss. And then by chance, as it often happens, so that's when I first came into contact with Carbotrace. And then we've taken up contact with Ebba Biotech. They sent us some Carbotrace samples to try, and later on we purchased our Carbotrace when we saw that it was working. And so I managed to give it a shot and try it, and it really worked out super well. So here's kind of like the introduction in how we proceeded to use Carbotrace to really help us solve the problems that we were facing during our cellulose research. So what we've been doing, we've been taking our cellulose samples, we've been taking the Carbotrace, we've been basically mixing them together and then use a confocal laser microscope. So that's the piece of equipment that we've been using. I've been very fortunate that we've got a very big chemistry department, and they've got lots of ties with biology. So we don't have any spectroscopy or microscopy ourselves, but luckily we were able to be collaborating with the biology department to use their confocal microscope for our research. Basically, this is what the Carbotrace website, the Ebba Biotech website, gives you. You've got the bound and the unbound cellulose, and you've got the different wavelength profiles. And basically here, I've been trying to replicate it. So the black line would be the Carbotrace only. So it would just be, just the simple Carbotrace unbound on the microscope. The red one would be the bound Carbotrace with the cellulose. And the blue one would be the autofluorescence of the sample. So every sample that you put on a confocal laser microscope, when you irradiate it with a special frequency, gives off an autofluorescence. So this autofluorescence you can see down here. So this is basically just my cellulose without anything put in the microscope, irradiated with a specific wavelength. And this is the autofluorescence that you get, and you get this red color. And you can see here that the autofluorescence of our sample does not interfere with the fluorescence of the Carbotrace. At first, we selected a Carbotrace by chance, which was identical with our autofluorescence. So we couldn't really take the two apart and tell which is the sample autofluorescence, which is the Carbotrace. But luckily, when we contacted Ebba Biotech, they've got lots of different Carbotraces which work at lots of different wavelengths. So you can really choose the Carbotrace which works specifically best for your sample and your specific situations. So that was quite good. What we then did was take this Carbotrace, mix it with our cellulose sample, and look at it under the microscope. And then we wanted to make sure that the Carbotrace really only flagged up cellulose, because we were suspecting there was still lots of hemicellulose, xylan in the sample as well. We wanted to make sure that the Carbotrace doesn't give us a response for both, a little bit of cellulose, a little bit of xylan. That wouldn't help us. We really needed it to be 100% specific just for cellulose. So that you can see up there, basically on the right side is pure xylan mixed with Carbotrace under the microscope, and you can see it's just blackness. So there's no response at all. So the Carbotrace, even though it's in there, doesn't bind to the xylan and doesn't show you any response. However, upon using pure cellulose, you can see we've got lots of bright green, beautiful response. So you can see that cellulose mixed with Carbotrace gives you a bright green response, which is great because it confirms for us that the only thing that the Carbotrace is showing us is the cellulose. And also down here, it doesn't really, the autofluorescence gives us a red response and the cellulose a green response. So they're very easily to be told apart unless you're colorblind. But that's an easy fix because the microscope can set the colors to whatever you want. So we just chose green and red for the sake of purpose and the sake of contrast. So then we used our own samples, and I used my different samples that I treated at different temperatures. So we've got the 160, 180, 200, and 220. Again, spray and spent, just two different types of microalgae. The temperatures and processes are all the same. And now the interesting thing was we mixed all of the samples with the Carbotrace, and we looked at them under the microscope, and we were very eager to find some sort of pattern, some sort of trend which would emerge and help us with our research. And what we saw was really better than we expected. So you can see there from the top right to the bottom right, from the top left to the bottom right, we are basically proceeding up in temperature. And as we go up in temperature from 160 to 180 to 200 to 220, similarly on the right side, you can see that the amount of green increases and the amount of red decreases. So the amount of red would signify that it's something which does not bind to cellulose, which means that, which is something that doesn't bind to Carbotrace, so it's not cellulose. So it might be xylan, it might be hemicellulose, it might be algaenan, which is in there. It could be in other cases, it could also be lignin, which isn't present in microalgae, but if you've got terrestrial biomass, it could also be lignin. And as we increase the temperature, you can see there's an increased amount of green, and the increased amount of green would show us that we're purer and purer in cellulose in our samples, and we wash out all the other stuff that we don't want more and more as we increase in temperature. And that also corresponds beautifully to the findings that we had before, where the only possible hydrogel could only be generated using those 220-degree treated samples. So, making the link between the hydrogel and the cellulose in the samples, we were able to do that through the Carbotrace, because the Carbotrace showed us the more cellulose we have in the sample, the better we can form hydrogels, and the other way around, we can form better hydrogels because we have more cellulose in our samples. And beyond just the fact of seeing more cellulose, of mapping that we have more cellulose, we can also see the two-dimensional spatial distribution of the cellulose in the sample. So you can see the different microalgal cells clumping together, the different pieces of sample and the different grains of cellulose that we've got in there. And you can see that the cellulose is pretty much spread pretty uniformly across the whole sample. We've got some clumps of cellulose and some clumps of red, like you can see down here, which is probably clumped together pretty well. So it might be more difficult to really flush it out during the solvent washes, but you can equally see some lumps of cellulose which really stick together. But especially down here at the MFC 220, you can see pretty well that it's a very uniform distribution of the cellulose, which really helped us to understand more about our samples, where the cellulose really is, what it really looks like. Especially if you zoom in, you can really gain a lot from it. And then we've also done some really beautiful image. I used them as the cover images in my PhD thesis because they're really, really nice. Basically, what you're seeing here is the initial microalgal cell. So in addition to just the cellulose, which we combined with the Carbotrace, we also used the microalgal cells fresh from harvesting, and we mixed them with the Carbotrace and we put them under the microscope. And you can really beautifully see, firstly, the round microalgal cells. So each of these circles, they're basically actually these spheres, because it's a three-dimensional, but we're only seeing the 2D section. But basically every single circle here is one microalgal cell. And in the right picture, you can see we removed the autofluorescence, the red channel, so you're only left with the Carbotrace cellulose channel. And you can really beautifully see the green rings which sit around the cell, which obviously corresponds to the cellulose being present in the cell wall of the microalgal cells. So you can really see beautifully where the cellulose sits in the microalgae, that it sits around the cell in a circle surrounding it. and protecting it as well, which is very beautiful because, you know, every species is different and lots of microalgal species are under-researched. And this is a proprietary species from Algaecytes. So nobody's had a look at those before. So they, you know, you really don't know what they look like until you look at them. And that was the first time that anybody looked at these microalgae. So it was a very beautiful, proud moment for us to see it, and really happy that Carbotrace gave us the opportunity to take those beautiful images. After we figured out the cellulose problem, we were still left with a second problem, which is the algaenan problem. So compared to terrestrial biomass, where cellulose is one of your main components of your cell wall, lots of microalgae also contain the curious case of algaenan, the curious polymer, which is called algaenan, and which not many people know about, basically. On the right-hand side, you've got basically cross-sections of different microalgal cell walls. And you can see that in there, we've got a fibrillar cell wall. So these hexagonal fibers here, they are basically lots of different carbohydrates, so mainly cellulose, for example, in the inside of the cell wall. And then we've got the algaenan-based layer, especially in chlorella, which you can see up here, which sits at the outside of the cell wall. So basically the part of the cell which is in contact with the environment. In this microalgal cell here, you can see it as well. They are sitting above the cellulose structure, and they're basically protecting it further from influences. So you have to imagine that these are unicellular species. So they are floating around in the ocean, they're floating around in lakes or anywhere. And it's just a singular cell. So it has to be very robust, and it has to protect itself from anything in the environment. And that's why this algaenan is very, very strong, very, very tough. It is basically acid resistant, heat resistant, alkali resistant. It pretty much resists everything. So that's why it's so difficult to crack, and that's why it's so difficult to also study. Up here, you can see kind of a generic molecular structure of this algaenan. The structure varies from species to species. Just because it exists as type A in chlorella doesn't mean that it exists in the same way in the algae that I used. So it makes it a very difficult thing to work with. You can see it's very carbon hydrogen based. So it's got a strong carbon hydrogen backbone. It's oftentimes unsaturated with some saturation in there, some double bonds, some ether linkages, some ester groups sticking out. So it's really highly cross-linked and very, very strong and very, very stable. And we were suggesting that that algaenan was also spoiling our hydrogels and also spoiling our cellulose. So it could explain some of the mystery peaks that we had in the solid state NMR. So what I did, very unconventional and probably not very green coming from a sustainable chemistry laboratory, but I basically soaked my cellulose samples in 2 weeks in concentrated phosphoric acid to really destroy everything in there and really get to that algaenan layer. And when I managed to filter it, which is horrible because phosphoric acid in its concentrated form is very viscous and you need lots of water to filter it properly. Really not a green chapter in my PhD, but it had to be done. Anyway, what we got was this kind of pure, what we hoped was algaenan sample. And I ran some more solid state NMR on it, and we could really see that the algaenan peaks, especially here around 30 and 40 ppm, were very much greatly enhanced and were basically more intense than the cellulose peaks that you could see in the middle, apart from this sample here. But in the top sample there, you can really see that the algaenan peaks were much higher than the cellulose peaks, which was, just as a quick reminder, back here was the other way around. You had your cellulose peaks being much, much more intense than the algaenan peaks. So we did manage, we think, to purify the algaenan, but obviously we had to reconfirm it, you know. We weren't really sure that all the cellulose and the hemicellulose was flushed out. We could still see from the solid state NMR that there was some residual cellulose, but we really needed another visual clue and another visual representation. So we used the Carbotrace again, and luckily, as we checked before, the Carbotrace really is only specific to the cellulose and not to anything else. We checked it with some pure algaenan samples as well, and we didn't really get any response. And then we were confident that we could apply it to these newly purified algaenan samples and really have a look, what's the cellulose in there like? Do we still have some cellulose? What does it look like? And basically what you can see here is mainly black which is pretty good for us. It's a very rare case where we're excited not to get any response from microscopy or spectroscopy at all. Usually we like to see lots of colors and lots of nice responses. But in this case, we were very happy about total blackness, because the total blackness would basically tell us we have no cellulose in there, and we were successful at removing every cellulose that we had left to really afford our pure algaenan. You can see there's still a little bit of a response. If you can see it well, I'm not sure how well you can, but basically here in the 220 spent sample, you can see some green that's left in the blackness, which is basically showing us that there's still some cellulose left, which also corresponds very nicely to this middle NMR spectrum here, which corresponds to that sample, which still has quite a lot of cellulose response. So, you know, we've got that connection again between the solid state NMR and the Carbotrace, which kind of links together really well and works as a tandem so well at explaining the research and the results that we've got in front of us. So basically, having confirmed that we are left with pretty much pure algaenan, you know, that then gave us the chance to really research the algaenan in more depth and really try to kind of crack the code of what the algaenan really consists of. And that gave us that great chance. Otherwise, you could do the IR, you can do the other different methods on it, like TEM, and you can never be sure, is what we're looking at really algaenan, or is it some cellulose that's kind of half still left? But because we were using the Carbotrace, we could really be sure that kind of 80% of what we're having in the sample is the actual algaenan. So it really, really did help us a lot, this Carbotrace, in advancing our research, drawing conclusions, and going further from it. So we were very happy and grateful to have found it. And it's been quite easy to work with. Obviously, it takes a little bit of a little while to really get into it, to understand everything, all the intricacies of the Carbotrace, and you know, you can really adapt it to your own needs. You know, you've got lots of different products that you can choose from, and you can really try and match it to what you're trying to do in your specific sample. So it was quite good to work with. And also, now that we've got this Carbotrace experience, I've been handing it over to PhD students coming after me. So we're trying to incorporate it into our research kind of standard research group methodology, which every research student working on biomass and celluloses can use. So it's pretty good to kind of have it established as a kind of ongoing methodology in our research group. So basically, that's the quick overview that I wanted to give you over my research, over my biorefinery research, and over the specific use of Carbotrace, and how Carbotrace, in my case, has really helped me continue my research and also elevate the quality of my research. And I'm leaving you with a beautiful picture from Algaecytes. They're the tubular reactors where the microalgae are grown. It's the beautiful pink light. When you leave the room, everything that you're looking at is green because your eyes get so adjusted to the pink light. Quite of an experience. So yeah, fascinating topic, and 5 years well spent. And yeah, thanks to Ebba for the Carbotrace and also for the opportunity of the webinar.